This last video will focus on amino acids. Amino acids are important because they are the building blocks of proteins. So if we think about it, a protein is just an amino acid polymer, or several amino acids linked together to make a protein with a biological function. So this video is mainly going to focus on the individual amino acids. Now the term amino acid comes from the fact that amino acids contain an amino group, or an NH2 group here, and a carboxyl group, or carboxylic acid. So the term amino acid, amino, and acid. The most common type of amino acids are alpha amino acids, and that's when we have the amino group and the carboxyl group separated by a single carbon. Now this is technically an alpha carbon because it's the first carbon after a carbonyl group. So here we can see that we have the amino group and the carboxyl group attached to the same alpha carbon. Now these alpha carbons are usually chiral centers for all of the different types of amino acids. That's because they're attached to 4 different things. So they're attached to an amino group, carboxyl group, an H, and then some sort of R group. Now the only non-chiral amino acid is glycine, and that's when the R group is replaced with a hydrogen. So since the carbon would be attached to 2 of the same things, it would make it achiral. A peptide is a term for a relatively short amino acid chain. By short, we mean about 40 to 50, less than 40 to 50 amino acids that are linked together. Now we can use prefixes such as di, tri, tetra to describe a peptide of a specific length. So for example, if we have 2 amino acids linked together, that would be known as a dipeptide. 3 would be known as a tripeptide. 4 would be tetrapeptide, etc. Or alternatively, we can use the term polypeptide. And polypeptide refers to a chain that's fewer than 40 to 50 amino acids. But this term poly just means many. It doesn't specify the exact number of amino acids that are linked together. Now, peptide forms when amino groups react with the carboxyl groups of different amino acids. So for instance, here we have 3 amino acids that are linked together. So we can see that the carboxyl group of one amino acid has reacted with the amino group of the other. As a result, we form an amide bond. The same thing can happen, and we can link these consistently. So here we have an example of 3 amino acids linked together, and this is known as a tripeptide. Now, as you link more and more amino acids and make longer and longer chains, the larger chains will eventually turn into things called proteins. There are 20 alpha amino acids that occur naturally. The thing that differs with all of these amino acids is the identity of their side chains, or the R groups attached to the alpha carbon. Now, all of the natural amino acids are L amino acids, and that's because their stereochemistry resembles L sugars, similar to glyceraldehyde, where the OH is on the left-hand side. So recall in the carbohydrates lecture that an L configuration corresponds to the S configuration at the chiral center. So here we have 2 examples of amino acids. L-alanine, where the R group is a methyl group, and L-serine, where the R group is a CH2OH group. So everything else about these amino acids is the same except for the identity of their side chains. Now these are both in their natural state, the L configuration, and we can verify this using the Fisher projection of each of the amino acids. So let's go ahead and determine our configuration and make sure that it is the S configuration. So for our alanine, the first group, or the first priority attached to our chiral center, is going to be the amino group. We know that our 4th priority is the hydrogen. Then we have 2 carbons attached, so we want to look further. This carbon at the top is attached to an oxygen and double bound to another oxygen, whereas the carbon on the bottom is only bound to 3 hydrogens. So this one would get priority number 2, this would get priority number 3. So we'll trace our path from 1 to 2 to 3. We're currently going in the R direction, but because our 4th priority is on a horizontal bond, which in a Fisher projection means it's a wedge, means we actually have the S configuration. Now, it's interesting to note that carbohydrates naturally occur in the D configuration, whereas amino acids naturally occur in the L configuration. Now, the 20 naturally occurring amino acids are also known as essential amino acids. Now, our bodies can make about 10 of those 20 amino acids, which means we have to get the other 10 from our diet. So meat eaters can actually get all of their essential amino acids from meat, fish, milk, and eggs. Now, for vegetarians and vegans, they're going to need a specific combination of grains, beans, and nuts. And there's no single plant source that can contain all of the essential amino acids. So you do have to have a varied diet in order to get the full 20 that we need. Now, one of the most important properties of amino acids is their acid-base properties. Now, since amino acids contain a carboxylic acid functional group as well as an amine functional group, we're going to combine the acid-base properties that we learned in the carboxylic acids chapter as well as the amines chapter. Now, with carboxylic acids, we learned that a carboxylic acid can either be protonated, or in higher pHs, this H can be removed and will form the carboxylate ion. With amines, we know that amines can exist as just a neutral state where the nitrogen is bound to two hydrogens, or it can be protonated to form an ammonium ion. So we're going to be focusing on the carboxylic acid either protonated or deprotonated, as well as the amine group either in its ammonium ion state or its regular amino state. Now, under acidic conditions, which means a very low pH, both the amino group and the carboxyl groups are protonated. Okay, so there's lots of protons around, which means both of these groups will be protonated. The acid will not have lost its hydrogen, its acidic proton, and the amine, which we know is a base, will act as a base and take the protons that are available. Now, we learned in previous chapters that our carboxylic acids and our ammonium ions have specific pKa values. So a carboxylic acid has a value of around 4.75. Our ammonium ions have values of about 9 to 11. So depending on the pH of the solution, we can have different versions of our amino acid. At a low pH, this is known as a cationic state for our amino acid. That is when we have both the amino group and the carboxylic acid protonated. Now, as we increase the pH to above the pKa of the carboxylic acid, eventually the pH is going to be so basic that this H is going to be removed to form the carboxylate ion. Now, at this point, we have the carboxylate ion and the ammonium ion. This is known as the zwitterionic state of our amino acid. Then if we continue to increase the pH to above the pKa of our ammonium ion, again, this is going to be a very basic state, and eventually this acidic hydrogen is going to be lost. This is going to form the anionic state of our amino acid, where the carboxylate ion is negatively charged and the amine is neutral. So overall, we have a negative charge, which corresponds to the anionic term. So there's a table in your book that lists the pKa values for all of the amino acids. Now notice that for the carboxylic acids in an amino acid, the pKa's are actually lower than a typical carboxylic acid. So instead of having a pKa of around 4.75, it's going to be closer to 1 and 2. Now this alpha COOH is referring to the carboxylic acid attached to the alpha carbon. So this pKa will correspond to this blue hydrogen here. The alpha NH3 plus is the ammonium, the protonated version of the amino portion attached to the alpha carbon. So again, this pKa value is going to correspond to the red hydrogen. Now, depending on the amino acid, the side chain can also have acid and base properties, and if it does, it's going to have a specific value as well. So for some amino acids, the R group itself can also have acidic or basic properties. Now for all of our amino acids, the carboxylic acid is more acidic than the ammonium ion. So as you increase the pH of a solution containing an amino acid, the carboxylic acid will always be deprotonated first. And this slide is simply showing the rest of the table with the pKa values of the rest of the amino acids. So the way that we're going to use pKa values when evaluating amino acids is to think about the definition of what a pKa is. So a pKa, by definition, is the pH at which the protonated version of a molecule and the deprotonated version exists at 50-50. So for example, the carboxyl group of alanine has a pKa of 2.34. So when the pH of the solution that alanine is in is equal to 2.34, the amount of the protonated carboxyl group, so this version, and deprotonated, so the carboxylate ion here, will be exactly equal. Now, as you increase the pH of the solution and get it more basic, you're going to increase the concentration of the carboxylate ion. So at physiological pH, which is about 7.4, since it's significantly more basic solution than 2.34, we're going to have a significant amount of the carboxylate ions. So at all pHs higher than 2.34, which is the pKa of this carboxylate ion, or of the carboxylic acid, more of the alanine molecules will be deprotonated. And vice versa. At pHs lower than 2.34, we're going to have a higher concentration of the protonated carboxylic acid. So for both carboxyl group and amino groups, we're going to see them protonated at pHs lower than their pKas and deprotonated at pHs that are higher. So let's go ahead and go through a practice problem. We're going to be drawing the structures of lysine at a pH of 1, 7.4, which is physiological pH, 10, and 12. So we're going to draw 4 different structures of lysine. So the first thing we want to do is take a look at the pKa values of lysine as well as the structure of lysine. So using our table, we identify the amino acid lysine. This is the structure of lysine. So it contains an alpha carboxylic acid, an alpha amino group, as well as a side chain that also has an amino group. So this is called a basic side chain. So the side chain itself will have a pKa value as well. So these are our pKa values of all of our groups attached to our amino acid. So again, the alpha carboxylic acid, which has a pKa of 2.18, we're referring to this hydrogen here. For the amino group, when we're talking about alpha amino NH3 plus having a pKa of 8.95, we're really talking about the H that would be attached to the nitrogen if it was protonated. And then for the side chain, it's a similar structures with the amino group. So the side chain, this pKa value of 10.53 is really talking about the H that would be attached to the amino group to make it an ammonium ion. So now let's go ahead and focus on drawing the structure of lysine at these various pHs. So a pH of 1 indicates highly acidic conditions. And since the pH of 1 is lower than all of the pKa's of all of the groups, it means that all of these groups will be protonated. So we can go ahead and draw our structure. So our nitrogen in the side chain will have 3 hydrogens attached, which will give it a positive charge. The carboxylic acid will remain with its acidic proton, and the alpha amino group will also have an extra proton and will exist in its ammonium ion state. So this is a cationic version of lysine, which exists at pH of 1. At a pH of 7.4, which is physiological pH, 7.4 is above the pKa of our carboxylic acid, but below the pKa's of our amino groups. So what this means is the carboxylic acid is going to be deprotonated, but the amino groups are going to remain protonated. So we'll go ahead and draw our structure at pH of 7.4. So since we have still acidic conditions with respect to the amino groups will be protonated. But they're basic conditions with respect to the carboxylic acid. So the carboxylic acid will lose its acidic proton. Now let's go ahead and focus on a pH of 10. At a pH of 10, we are now above the pKa's for the alpha carboxyl group as well as the alpha amino group, but we are still lower than the pKa for the side chain. So since we're a higher pH than the pKa's of these groups, these will all be deprotonated. However, the side chain will remain protonated. So go ahead and draw our side chain first. Then we will draw our alpha carboxyl group deprotonated with a negative charge, and the alpha amino group also deprotonated, so it's now going to be neutral as well. And then finally, we'll focus on the pH of 12. Now at a pH of 12, we're higher than the pKa of all of the groups. Sorry, a higher pH. That means all of our groups will be in their deprotonated forms. So we'll draw both amino groups, neutral with 2 hydrogens, and our carboxyl group will also not have its acidic hydrogen. So this, because it's a negatively charged version of our lysine, this is the anionic form. Now at physiological pH, the structure of an amino acid is zwitterionic. That means that the carboxylic acid is negatively charged and the amino group is positively charged. So even though we have opposite charges, overall it is a neutral state for this amino acid. Now the reason that it's going to exist zwitterionic at the physiological pH is because physiological pH, which is around 7.4, is higher than the pKa's of all of the alpha carboxylic acids and lower than the pKa's of the alpha ammonium ions. Now the isoelectric point, also known as the pI, is the pH at which the zwitterion concentration is the highest. Now it may be close to, but not exactly to, physiological pH, depending on the identity of the amino acid. Now to calculate the pI for an amino acid, and these are specifically the instructions for calculating it when it doesn't have a basic or acidic side group, you're simply going to average the two pKa values. So for instance, in this amino acid, the R group is just a regular alkyl group or a group that doesn't have acidic or basic properties, so there's no pKa value for the side chain. So what we're going to do is we're going to take the pKa value for the carboxylic acid and the pKa value for the ammonium, and we're simply going to average it. So for this molecule, the pI is 6.02. So if you were to place this amino acid into a solution that has a pH of 6.02, we would have the highest concentration of zwitterion. For amino acids that contain acidic or basic side chains, what we're going to do is average the pKa value of the side chain with the matching amino or acid group. So what that means is for acidic amino acids, meaning amino acids that have an acidic side chain, such as glutamic acid, we're going to average the pKa of the side chain, which is 4.25, with the pKa of the alpha carboxylic acid, which in this case is 2.19. So averaging these pKa's, we get a pI of 3.22. So for glutamic acid, this is the pH at which the Zwitter ion would be the highest. For basic amino acids, we average the pKa value of the side chain with the ammonium group. So lysine, which is the example we just did, contains a basic side chain since it's an amino group. So we will take the pKa value for lysine, which is 10.53, and average it with the pKa for the ammonium group, which is 8.95. So for lysine, the highest concentration of Zwitter ion is going to be at a pH of 9.74. Now, peptide synthesis is a very common type of synthesis in pharmaceuticals, and that's because if you can make a drug that consists of amino acids, it's less likely to be rejected by the body. Now, let's consider the process of peptide synthesis. Now, we know that in order to form a peptide or an amide bond, we need to have the amino group of one amino acid react with the carboxylic acid of a second amino acid. Now, if we're working with two amino acids here, there's the potential that we can get 4 different dipeptides. This is very similar to the cross-aldol reaction. So we could have the amino group of the second amino acid react with the carboxylic acid of the first to get our first dipeptide. We could have the amino group of the first amino acid react with the carboxylic acid of the second to get our second dipeptide. We could have 2 of the second amino acids react with each other to get the third dipeptide. Or we could have 2 of the first amino acids react with each other to get our fourth. So if we have a specific way of how the peptide needs to be assembled, we can use protecting groups to kind of guide the amino acids to react in a specific way. In order to force amino acids to couple together in a very specific way, we can use protecting groups. And specifically, we're going to protect the N-terminus or amino group of one carboxylic acid and the C-terminus or carboxyl group of the other amino acid. After protecting them, we leave one carboxylic acid group available to react with one amino group. Once these are ready, we will perform a reaction with what's known as coupling reagents. So we learned in our carboxylic acids chapter that carboxylic acids are not very reactive. So they need a little bit of help in order to form our amide bond. So that's what this reagent is here, DCC. It's called a coupling reagent, and it essentially makes the carboxylic acid significantly more reactive to ultimately react with the amino group. Now, after they've reacted with each other to form the amide bond, which is here, we've coupled our 2 amino acids together, we have formed our dipeptide, and then at this point, we can use reactions to remove the protecting groups to get our desired dipeptide. So peptide synthesis is the way that chemists use to form small chains of amino acids. Biologically, nature will put super long chains of amino acids together to form proteins. Now, there are 4 levels of protein structure, which you'll probably cover in biology or a biochemistry course. But we have primary, secondary, tertiary, and quaternary structures. And these are the 4 levels that are needed to describe the three-dimensional shape of a protein. The primary structure of a protein is simply the amino acid sequence. So it tells us all of the amino acids that are coupled together with amide bonds. The secondary structure refers to the 3-dimensional shape of very localized regions along the protein. Now, these types of structures are mainly dictated by hydrogen bonding interactions along the peptide backbone. So hydrogen bonding can exist because we have oxygens, we have nitrogens with hydrogens attached. We also can have side chains that have hydrogen bonding capabilities as well. So those different types of interactions are going to dictate the secondary structure. The tertiary structure of a protein refers to the three-dimensional shape of the entire peptide chain. Now, the shape of a protein can actually change depending on the environment that it's placed in. So, for example, if it's in a polar environment, the protein can actually adopt a shape that puts the polar groups around the exterior. And that's because like interacts with like. The polar groups will want to interact with the polar environment. In a nonpolar environment, the polar groups will be in the interior and the nonpolar groups will be around the exterior. Again, like interacts with like. The term denaturation is when the intermolecular forces, or the forces within the protein that hold it together, are disrupted. Now, this can occur in extreme temperatures, in different pHs, etc. So the proteins are very particular about the environments that they are placed in. The quaternary structure is when multiple peptide chains come together and interact with each other to ultimately form a protein. So for example, hemoglobin is made up of 4 peptide chains that all interact with each other to form one structure that has a specific function. So amino acids play a very important role in biological systems.